So, welcome, a very warm welcome to those of you in the room here in Southampton, and also a warm welcome to those of you joining us for this event, loosely to commemorate World Cancer Day, which is actually next week, but part of our series of talks and events around the theme of cancer and cancer immunology. My name is Mark Spearing, and I have the honour of being the Vice President for Research and Enterprise here at the University of Southampton and your host for this event this evening. Part of that honour is I get to give the housekeeping notices for those in the room. We are not expecting a fire alarm, but were there to be a fire alarm, it would be for real. Please take the exits either side, best flight attendant, follow University of Southampton staff members down the stairs and gather at the assembly point, which is just across Salisbury Road in the car park opposite the front of the building. For those of you online, you don't need to worry about that. I will shortly be handing over to Mark Cragg, who together with Gareth Griffiths and Esme Fowkes work in our Centre for Cancer Immunology, and we're also delighted to be joined by Ali Richards, a patient who has participated in trials around cancer vaccines and cancer immunology. And Mark will give a broader introduction to our speakers this evening. But what I'd like you to do, as you listen to our speakers, is to think of questions, and we'll be allowing about 20 minutes at the end for questions, and particularly those of you online, you should see in front of you on the screen a box in which you can enter questions, and I will do my best in comparing the Q&A session to make sure we have at least as many questions from online as those from in the room. In the room you'll have a microphone that will come to you if you want to ask a question, so those online can hear the question you're asking. So that completes my introduction. Again, welcome to everyone. I'll now introduce Professor Mark Cragg, who will then introduce our three speakers. Mark, over to you.
Fantastic. Thanks very much, Mark. So it's my job really to give you a very brief 5-minute introduction really to the theme, the scientific theme of what we're going to talk about. And so really that starts with a couple of definitions. So all of the things we work on in the Centre for Cancer Immunology are cancer immunology. And what is that? So that's the study of how cancer interacts with the immune system. And it's really important that over the last 2 decades, probably, we've got a much greater understanding that cancer isn't just cancer cells on their own, but it's actually cancer cells talking to and being talked at by our own host immune cells. And that's a really key fundamental thing that we've come to understand and is probably the basis for the centre in itself. The other key term that you'll hear about and the kind of thing we focus on in the centre is immunotherapy. And that's using the immune system to treat disease. And over the next couple of minutes, I'll kind of give you a refresher, really, of what we're trying to do and why we try to do the kind of things we do in the centre. And really, it stems from this kind of interaction that we see between cancer cells and immune cells. So we have this concept of cancer immunosurveillance, which basically shows that throughout your body all the time, cells are becoming mutated, they could be cancerous. The immune system is clever enough to detect that and destroy it before it becomes a problem. However, what happens in certain cases is that the cancer escapes. It is able in some way to evade or suppress the immune system, which is why we then get to a point where we get clinical presentations of cancer. So that's the problem that we face, and I think prior to probably 2000, people didn't really appreciate that it was that subversion of the immune system that allowed cancers to develop. But that also provides us with a recognition that if we can re-engage immunosurveillance, if we can switch back on those immune cells or boost responses more favorably using immunotherapy, we can then start to get cancers to regress again. And that's, in essence, what we try to do in the centre. But as I'm sure you all would appreciate, this is a very complicated process. There's lots of cells involved, there's lots of molecules involved. And so to really understand this process, we need a critical mass of scientists to really tackle this problem. And so that was really the birth of the centre. That's why it was formed, was just to bring a critical mass of people working on cancer immunology to allow us to design new immunotherapies. But I think a key concept was that this isn't just about us understanding something. We need to then be able to take that understanding and translate it into the clinic to get better treatments for patients. So that's why in the centre we have scientists, clinicians, and clinical trialists actually taking our findings into the clinic so we can test them to get better treatments. And that's a really important thing of what the centre does. When we think about how we're going to do that, really we take all of our knowledge of immunotherapy, how the immune system interacts with cancer, and all of the things around this slide. If anyone's been to the centre and heard me talk, you will have seen this slide. I use it every time because it really just summarises everything that we can do to try and fight against cancer. So some of these things, if you came and heard me talk specifically, it would definitely be about antibodies. I think that's one of the most effective ways of developing new treatments. But actually, what we're going to hear about tonight really is as another concept, which is how we can use vaccination to fight against cancer. And this is a particularly big area at the minute. People are very excited about the idea that we could do this. And there's lots of different ways that we can do this. People have been trying different types of vaccinations over the last few years. And now particularly, and some of the things you'll hear about from Gareth is the things that are actually going into the clinic, is trialing things like DNA and RNA vaccines. So these are the things that we're actually doing now. Obviously, back in the lab, we're trying to design more effective ways of vaccination. And the thing that you'll hear about from Esme is not vaccinating about some differences that might be in a tumor cell, which is the kind of DNA, RNA vaccine strategy, but actually trying to boost the immune cells themselves so that we get better responses in terms of vaccinating against cancer. But you're probably already aware that we already use vaccination in the treatment of cancer. And this is where we're vaccinating against viruses. So HPV is responsible for a large amount of cervical cancer. We already have a vaccination strategy there that's been very, very successful. The same for vaccines against hepatitis B, which is a leading cause of liver cancer. Again, these viruses are known to contribute to the development of those cancers. So if we can stop the virus, then those cancers don't develop at the same rate. But what we're talking about tonight really is an alternative strategy, was either vaccinating against the cancer itself, or as I mentioned, what Esme will talk about is actually boosting the immune system to get better vaccination against cancer. So that's what we're going to hear about. Gareth is going to take you through some of the clinical trials that they performed in this area of vaccination. Then Esme is going to talk about some of these novel ways that we might manipulate the immune cells to get better vaccination strategies. And then Ali's going to give you her personal perspective of being involved in some of those trials. So without further ado, I'll invite Gareth to come up. So he's our director of the CR UK Clinical Trials Unit, and he's going to give you a deep dive into cancer vaccines. Thanks very much.
Thank you very much, Mark, for that introduction. So, I'm Gareth Griffiths. I'm a professor of clinical trials. So I'm a mathematician, statistician by background. And I'm based within the Centre for Cancer Immunology, as Mark mentioned. Absolutely fantastic building. Upstairs, they'll have Mark and the scientists working in the laboratory. And I'm on the ground floor. And although we're all University of Southampton, we're based up at Southampton General Hospital. And that allows us to work very, very closely with the hospital and their patients to find evidence for new studies that work. And those that remember, the way I sort of describe it, those who remember years ago when we used to be in supermarkets and banks, we used to have that vacuum system where you used to put the money in and send it up. It used to go to the head floor and they used to stash away the money in the safe. And I almost see that as a reverse. Mark's upstairs and his colleagues trying to discover new treatments, and the idea comes down to us in the trials unit, and we develop a clinical trial. And what we try and do is all about creating evidence for change and patient benefit, in the hope that if we discover a new cancer vaccine or another treatment, the hospitals around the UK start using it. So we're like a data centre. We don't see the patients. We interact with all the hospitals in Southampton and around the UK to recruit into our studies, and us mathematicians tell us whether beyond reasonable doubt something works and should be used as standard of care in the NHS. And what we do is we work from the very, very basic idea. We're very outward facing. Mark and colleagues will come with ideas. Doctors in the hospitals treating patients will come with ideas. Sometimes patients come with ideas. And what we do is we develop that study, run it, collect data, look at the data, and statistically prove whether or not that treatment works. And what we do, we publish that in a high impact publication. And what we hope is that the NHS, through NICE, We'll look at our data and then say this should be given to every patient in the UK going forward. And that's what we're about, creating that evidence for change. But within those studies, we collect samples, such as cancer tissues and bloods, and we send them back to Mark. Because what we know is that sometimes we find a new cancer vaccine, and it works in the majority of people, but some people don't benefit, or some group will highly benefit, and we're saying, well, what's going along biologically? So we work very closely back and forth to discovery science. And we share that data as well, and that's so important. We learn across the world, because what we don't want to do in clinical trials is do a study which the Americans are doing or the Swiss are doing and just waste resources. We can share knowledge. So I'm going to give you some examples of some studies we're running within the Southampton Clinical Trials Unit. So Mark mentioned human papillomavirus. We all know we vaccinate our 12-year-old girls because we know HPV is associated with cancer of the cervix later on. And what we know is that human papillomavirus, especially a strain which is HPV-16, is related to cancer in some cancer types. And we know that happens. And HPV is very, very common, and a lot of people pick it up. And the majority of us are able, your body's immune system is just to remove that HPV vaccination. But for some individuals, for some reason, their body can't remove the HPV vaccination, and it can cause cancer. So our first study I'm mentioning is where we're trying to target the human papillomavirus with a vaccine to remove it, so then it allows your body to fight the cancer. So there was a new cancer vaccine. It is called a messenger RNA vaccine, which is a new development of vaccines. They learnt a lot from COVID. And what they've said to us with the company is, right, we've got certain doses of this vaccine. So the first question we need to investigate is how much of a dose can we give to a patient? Because if we know if we can increase the amount of treatment a patient receives, the more potential is to be active, but we don't want to give too much treatment. Sometimes there's side effects, sometimes there's not. And we start off with a phase 1 study, so it's a first-in-human study. And what we do is we slowly escalate the dose, and I'll show you how we did that. And we started off, first of all, measuring immunological response. So we're taking samples from the patient all the time and saying, is this vaccine initiating the body to start fighting the cancer and remove the HPV? And then what we do is we move on to much larger studies, including a variety of patients, human papillomavirus. We started in head and neck. We included esophageal throat cancer, anal cancer, cancer of the penis, anyone that we know is associated with human papillomavirus, to work out whether that vaccine is a viable treatment. And we approach patients and we say, would you like to come to the study? And Ali's going to talk about that sort of process. And it's completely up to the patients whether they come in, and we have to collect lots of data. And there's lots of dosing in this study. And within this study, we kept on increasing the dose and looking very, very carefully. But an advantage working at Southampton General Hospital is there's a clinical research facility that has state-of-the-art to facilitate patients getting involved. And that's a really, that study's going to report later on this year. It's a really novel approach. So let's move on to another cancer, colorectal cancer. We know in the world, 1.2 million people will be diagnosed with colorectal cancer every year. We know treatments have had a massive benefit, but on average, 50% of people will die of colorectal cancer. So we've got to use new strategies to deal with colorectal cancer. And what happens with some cancers is they have this shield around them that prevents treatments from working, or your immune system from identifying the cancer. So we can use cancer vaccines to remove that shield. In this case, it's PD-1, so that suddenly your immune system can see the cancer and just kill it. So what we've done is we developed a study where we recruit patients, and I'm going to show you the process. We know in some subgroups of patients that have something called microsatellite instability high, we know they're supposed to be more susceptible to immunotherapy. So we select those patients. And we do a CT scan, see how the cancer is at that moment in time. We then give them this vaccine because we want to see what the impact is on the cancer. We then take another CT scan to see whether the cancer is shrinking as a consequence of this. And then we do surgery, which is standard of care, and trying to remove the cancer. Because sometimes if you get localized cancer like surgery, like colorectal cancer, you can use surgery to remove it, but you can't see the 1 or 2 bits of cancer that are still circulating around the body. And it can come back in a couple of years' time. So what this is doing is targeting those cancers and trying to kill it, so that once you've had your surgery that removes all the cancer you can see, you're less likely to come back. And we collect lots of data. We continue to collect samples, which go back in the lab with Mark to understand more about the cancer and the treatments. And we follow up over time to see what the impact is in overall survival and what happens with the cancer, but also quality of life. Because we don't want to give cancer vaccines to patients that give them benefit but makes them feel quite ill as a side effect. So we measure everything. And with this particular study, within the University of Southampton, we realised that colorectal cancer doesn't just happen in the UK. So we're actually, the University of Southampton is allowing us to open this up in Australia as well. So we're going to recruit patients from both the UK and Australia, and that could change practice both for the UK but also Australia, which is absolutely fantastic. Now, the third thing is there's been a massive development in cancer vaccines. You've heard about a couple of vaccines you don't really select for patients, but there's new state-of-the-art now where when a patient goes into hospital, and if they're having surgery and you can take their cancer out, you can send it off to a laboratory. They will look at the genetic makeup of that individual's cancer. They then make a cancer vaccine for that person and send it back for that person to take. And that happens, say, in colorectal cancer, like I mentioned. So if you could take out the cancer as part of your standard of care because you're having surgery, if you can just send a piece of that off to be analysed to create a cancer vaccine, send it back, and that patient has it, that it mops up all the little bits of cancer no one can see on a scan because they're single cell. That's an amazing development. The problem and the challenge is that a lot of our patients are seen not just in state-of-the-art hospitals like Southampton General Hospital that can do sophisticated analysis and translational. They're seen in district general hospitals. They're seen in hospitals all over the world. So we need to think about a mechanism in which we can recruit patients and then introduce them to state-of-the-art trials. And there is an NHS England government initiative to develop a platform which we're running in Southampton General Clinical Trials Unit, where basically we recruit patients, collect their samples, do a load of analysis, but introduce them to the state-of-the-art clinical trials that are happening. And some of these trials are outside of Southampton. And I'm just showing you how we do this. So the problem has been slow recruitment, because if we're going to select individual patients and everyone's different, it's not just everyone with colorectal cancer we're interested in. We're trying to break it down into a bit different genomic groups. Trials need to be run across the UK because we want to have an impact on all patients, just not those in state-of-the-art hospitals. And sometimes with samples, you have to process them in specific ways to get the genetic data out. So one hospital doesn't necessarily generate in another way. But as here, patient benefit's always the drive. So we need to set up a structure in the UK so that if we find evidence of benefit, the NHS across the whole of the UK is going to use it. And we were successful in running this. It's called the Cancer Vaccine Launchpad. And we're running that within Southampton Clinical Trials Unit. And essentially, we've started with colorectal cancer patients. These are patients which have a cancer and they're going to come in for surgery. And we approach the patient and say, would you like to come into our Cancer Vaccine Launchpad? If they say yes after reading information, we take a little sample, part of routine. We send it off for processing within the NHS. That's sent to a state-of-the-art laboratory where they do the genomic analysis. And if they're eligible for a study, we introduce that patient to where that trial is taking place, and they enter that study with precision therapeutics. So we're creating this system where all these blue centres are open, and if patients are eligible to go into a trial, which is a centre of the red spot, we can make that introduction and they can go into that study. And it's having an absolutely massive impact. And we're recruiting at the moment across England. We're expanding it into Scotland and Wales and Northern Ireland at the moment, where we're recruiting hundreds and hundreds of patients, profiling them and introducing them to state-of-the-art trials that are out there to make a difference. And it's had a massive impact. It's allowed us to screen a lot of patients. And actually, it's one of the things where we're beating the Americans and all the other countries, because when they've looked and opened these trials across multiple centres, the UK is recruiting 4 times as many patients as any other country. Absolutely fantastic, because actually we're working, together as a community across the NHS. And actually what we've done, because we're opening up in all those sites, like district general hospitals, not just state-of-the-art massive treatment hospitals, at the moment we're recruiting 61% of all colorectal patients, or we're screening the patients. So 61% of all those colorectal cancer patients every year in the UK, we're asking, would you like to be part of this? If they say no, fine. But if they're interested, their samples go off, processed, and then we introduce them to those studies, and it's making a massive difference. And the advantage of that is diversity as well, because we're quite a diverse population across the UK, and by opening it up across the UK, we can learn a lot more, so it's making an absolute massive impact. And that's just an example of some of our studies. Very early first-in-human studies targeting HPVs, studies of where we have international collaborations that could change practice, not just here in the UK, and studies in which we facilitate massive screening of patients and identifying those patients for the best trials for them. So thank you very much.
He told me he'd go long. Anyway, thanks very much, Gareth. And hopefully what he's been able to convey is the really 3 different types of vaccines. So the first one is addressing the virus. The second one actually involves an antibody that boosts the immune response. And then that last one, which is a highly personalized type of vaccine strategy, which, as you can see, takes huge logistics to actually set up and be able to implement. So that's really exciting to see how well that's going. So now we're going to shift gears a little bit. So we're going to move on to Esme. So Esme is one of our PhD students. She's entirely funded by philanthropy through the Cancer Immunology Fund. And she's been doing, just recently, in the last month, finished her PhD. She's going to tell you a little bit about some of the work she did in that PhD as it relates to vaccination. Okay, thanks very much, Esme.
All right. Yeah, thanks, Mark, for the introduction. So I'm going to speak a little bit today about my PhD in cancer immunology and some of the research that I've done looking at using STING agonists to boost vaccine responses. So I started my PhD in October 2020, which is certainly a slightly strange time to move to a new city and start a PhD. But a lot's changed in 4 years. I've really enjoyed my time here. And as Mark says, I've recently submitted my PhD thesis. And I've actually continued to stay working in the same lab in the Centre for Cancer Immunology for a few more months. So before Southampton, I was studying natural sciences at the University of Cambridge. So I've always loved science, really. And I think it was probably as early as my first year that I started thinking about doing a PhD. Really enjoyed doing hands-on research and being able to find out things. And in particular, I did a research project in immunology in my final year. And I decided that I wanted to do a PhD. So I came across the Cancer Immunology program, which is offered in Southampton. And I was fascinated by the concept of using our immune system to treat cancer and the breakthroughs that was beginning to make in treating some patients and the potential that it showed for treating many more patients. And of course, Southampton has been heavily involved in cancer immunology research. And at that time, the Centre for Cancer Immunology was only recently opened and specifically dedicated to this area of research. So I did my PhD within the antibody and vaccine group in the CCI, and this is one of this is the biggest research group in the CCI. There are a few. It's a little unusual in that it's composed of 7 or so principal investigators. So it's very big, and we all sort of have slightly different focuses of research, but day to day we all work together, and that creates a really collaborative and supportive environment, which is very fruitful. So to talk a little bit about my project, so as we've heard, I've been focusing on a protein called STING, which is a stimulator of interferon genes. So this is just one component of our immune system, but it's a protein which is present in a lot of our cells and particularly highly in our immune cells. And it can detect if our cells become infected with a virus. It does this by sensing DNA in the cytosol of our cells. So outside of the nucleus where DNA is normally stored. And when STING is activated, it sends like a warning signal to our innate immune cells. So these are sort of the first line of defense of our immune system, which then leads to the activation of T cells. And these are able to kill virally infected cells, but they're also crucial cells for killing cancer cells. And interestingly, STING can also be naturally activated within some tumours. Because cancer cells divide so uncontrollably, they can actually accumulate DNA in their cytosol outside of the nucleus, where it would be in healthy cells. And this is thought to contribute to the activation of T cells, which we see in some patients, and is generally associated with better responses to immunotherapies, better prognosis. So there's been interest in therapeutically targeting STING with drugs which can activate it. So these are called STING agonists or STING agonism. And this has the potential to promote T cell responses and drive anti-tumour immune responses in cancer patients. And this could potentially be used in combination with other immunotherapies to increase their efficacy. So there's been a fair bit of interest in STING agonists. Pharmaceutical companies have developed various different molecules which show potential to induce anti-tumour responses. And several of these have proceeded to clinical trials, but so far they haven't shown a lot of patient benefit. So my project has really been looking at better understanding how STING agonists lead to T cell responses. So firstly, I've looked at the effects of STING activation on our innate immune cells. So I've treated mouse and human cells with various STING agonists. These are the different coloured bars, and several of these have been evaluated in clinical trials. And we see clear concentration-dependent upregulation of markers of activation on these cells. So it's clear that STING agonists can activate our immune cells, but as I've said, they haven't had a lot of benefit in patients yet. So to look at how STING activation leads to T cell responses, I've performed vaccination studies. So to go into a bit more detail about vaccines, they comprise of an antigen, which leads to the activation of specific T cells. But in order to be activated, T cells need to be presented this by an activated innate immune cell. So vaccines also contain an adjuvant, which stimulates the innate immune cell activation to be able to activate T cells. And so we can use STING agonists as a vaccine adjuvant. And I performed these studies in mice. So mice are a really important tool for us to be able to study responses to vaccination. It's very difficult to study the sort of complex interactions of so many different immune cells in a Petri dish. And mice have STING, which is functionally very similar to human STING, but there are some crucial differences, and this can affect how much they're activated by some agonists. So in Southampton, we have a human STING genetically modified mouse, and hopefully this can allow us to better model how patients respond to STING agonists. So briefly, these mice are administered with antigen-specific T cells, which are not activated. And then the next day, they're vaccinated with an antigen and STING agonist as the adjuvant. And then through blood sampling, we can monitor the expansion of these antigen-specific cells in the blood. This is a technique called flow cytometry, which is commonly used in immunology to identify specific populations of cells. So by sampling over time, we can monitor their expansion, and we see that STING agonists lead to good expansion of T cells. So they rapidly expand, they peak at day 5, and then they will decline, but a small population of memory cells will persist. But interestingly, we found that actually giving higher doses of STING agonist led to a lower magnitude of T cell response. And it is quite common that actually overactivation of the immune system can cause the engagement of negative feedback mechanisms. And this is one of the ongoing challenges of a lot of immunotherapy, to drive sufficient but not overactivation of our immune responses. So I was able to investigate the potential sort of underlying mechanisms behind this. And I identified some conditions which could boost the response induced by higher doses of agonist. And actually one combination treatment which led to equal or even greater magnitude of response. And so these kind of studies give us a better understanding of the biology which is affecting how STING activation leads to T cell responses. And hopefully this can be applied in the future to develop better STING agonist immunotherapies. But a crucial question will be whether this can be used to boost T cell responses against cancer antigens and therefore drive cancer cell killing. And this hasn't taken into account the immunosuppressive tumour microenvironment, which is very common, and that is an ongoing challenge of various immunotherapeutic approaches. But ultimately, STING agonists could be used to boost T cell responses against cancer cells in patients. So I hope I've given you a bit of an insight into the potential as well as some of the challenges of using STING agonists as cancer immunotherapy. So thank you for your attention.
Fantastic, Esme, and on time. So I just wanted to go back over a couple of Esme's slides that I was listening to, just to highlight a couple of things. And really, the first of them is this. So looking at graphs, maybe you're not all used to looking at graphs every day, all day like me. But the black line is what you get with vaccination without the STING agonist. There's almost no response. And it's only when you add the STING agonist that you get this really fantastic response. So that's the first thing, the power of some of these agonists to get much better responses. And then the other thing to mention is that when we're vaccinating often thinking about cancer vaccination to get T cell responses, but we can also produce antibodies. And antibodies are actually very useful for fighting off viruses, infections. So those HPV vaccines we've talked about might actually be more important to get antibody responses than T cell responses. With these STING agonists, one of the things that Esme hasn't had time to talk about is we actually have also looked at the ability of these STING agonists to boost antibody responses. So there's a huge level of sophistication about thinking about what is it we need, what are the key determinants of a vaccine that will give us better anti-cancer responses. And we start off by looking at a reductionist approach, so we can look at a vaccine approach. This is actually to a foreign antigen from egg white. It's just what we use experimentally, but obviously then the plan is then to move it onto more and more sophisticated types of models. And the other thing that Esme mentioned was this idea of overcoming the immunosuppressive microenvironment. So I said right at the start, the only reason why those cancers develop in the first place is because they've evaded the immune system. They have suppressed it to allow them to grow. And so the idea that these STING agonists can actually reactivate the immune system is, again, very exciting to us. And that's one of the things that we're interested in developing. Okay, that's the key science part. So we're going to now move on for our third talk with Ali Richards. So Ali's going to basically give you the human angle on what it's like to be part of some of those trials and the experience of that. So thanks very much, Ali.
Hello, everyone. You've had the technical talk. There's no technical in this whatsoever. I can promise you that. So This is me. What this tells you, first of all, is I can't take selfies. But I took this picture when I was actually on the trial in the ward, because I was trying to emotionally blackmail my friends and family into helping me with some fundraising. And I found that little card, and I still have that little card today, because I'm really quite proud of what I hope I've contributed to this. So, selfies aside, let me tell you a bit about my story. So I found a sort of lump in my neck in Christmas 2015, thought, it's maybe a little cyst or perhaps it's a gland. Went to the GP, and because of my age, I got quickly referred through to Poole Hospital. And they were very quick. They gave me a diagnosis within the day. That's what the official diagnosis is. To us, to normals like me, I had throat cancer. And the cancer was on the back of my tongue, and it was caused by the HPV-16 virus, which Gareth mentioned earlier on. That's all very matter of fact. I went through loads of scans and tests. I had an operation to remove what was left of my tonsils. There was hardly anything there. And to take a biopsy. Now that biopsy will come back later on, because that was important. And then I had to go through things like restorative dentistry and audiology, all of this before I could start my treatment. But how I felt was a completely different thing. So I was on this track with the NHS, but at the same time, inside, I was absolutely petrified. My first question when I got my diagnosis was, am I going to die? And the answer was, that's not the plan. Thank heavens for that. I question myself a lot. I look after myself. So what had I done wrong? As it turns out, it was nothing. It was just bad luck. And as the guys have explained, I just couldn't fight this particular virus. But I was used to knowing stuff, being in control, knowing where my life was going. And that loss of control was really disconcerting. I felt angry. Why me? But that, I suppose a lot of people feel like that. But I also felt guilty about the impact it would have on my husband and my son. And I felt really isolated. We'd just moved to Poole from Nottingham, and so my whole support network were up there in Nottingham. So it was a really tough time for me and my husband, who'd just started a new job. So I started my treatment in March 2016. The first thing I had to have done was a peg fitted. That's a feeding tube in your stomach. And I was like, this seems a bit extreme. No, you might need it, I was told. I then had 5 chemotherapy treatments and 35 radiotherapy treatments. So every day for 7 weeks, I was pinned down to a table. That's me, that is. That's my radiotherapy mask. So they pin you down so they can make sure you're in the same place every time. It started out like a breeze. I thought, 2 weeks in, not a problem here. After that, it all went really quickly downhill because you've got the cumulative effects of the treatment. And I know you hear about clinical trials, but I just want to explain this a bit to you because I think it's important and it's what's motivated me. So short term, in the end, I couldn't speak. My husband was quite grateful, but it was problematic. I could barely whisper. I had biblical quantities of phlegm from the radiotherapy. All of this, I don't need to read it all to you, but I was in so much pain that I actually cried in the shower because the water hurt me. And I used to throw up every time I brushed my teeth just because of the way my body was reacting. I never felt ill from the cancer. It was the treatment that made me feel really ill. And I don't want you to think I'm moaning about the treatment because I know I had the very best treatment and care available from Poole Hospital. I know that. But it's brutal. And it's not just the short-term impact. I've got long-term impacts from this as well. And I lost confidence in myself and my body because of so much emotions going on with all of this as well. Just when I thought that I was getting out of the woods, I had a bit of a setback. although the treatment had got rid of my tumour on my tongue, it hadn't got clear of everything in my lymph nodes. So I ended up having to have a neck section. I couldn't do selfies. My husband had to take these because I wanted to know what it looked like. But I had yet more surgery. I also had to have physio and lymphedema treatment. And it's left me again with more nerve damage. So my arm doesn't work quite properly. Again, I'm not moaning, but I just want to explain it. So, into the new normal. You never go back to the old normal, I don't think. You go to the new normal. And what I wanted to do was something positive. And so I threw myself into a whole load of things. The fundraising I mentioned earlier. I did the 10,000 steps a day. I started volunteering for things. I had a complete career change because I'd had enough of my old career. This marvellous woman is Dr. Emma King. And she was my oncologist at Paul Hospital. But unbeknownst to me, she'd also talked to Gareth about, along with another colleague, about this idea of the throat vaccine. So it was her who contracted me into getting onto the trial. And as soon as she asked me, I said yes. Because I trusted her, but also because I didn't want other people to go through what I'd gone through with my treatment, which is why I was trying to explain that to you. What Emma explained was it could also help me, but my motivation was really to stop other people going through that. So I joined the trial in September 2018. It was funny looking at Gareth's slides because I was in that cohort too. I was one of those red, yellow, green boxes. It was just quite funny. So I checked in with the team, who were all very welcoming and kind. They gave me an assessment to ensure my suitability. They put a dose on my back. When I asked my husband to take a photo of my back with just my pants on, he thought his luck was in. He was wrong. It was my post-dose test. And I signed all the consent forms. And I'm going to be really honest with you. Did I understand everything? It was explained to me really well. But did I understand it? No, I did not. But I trusted in Emma and I trusted in the team. And I absolutely believed this was the right thing to do. So I went ahead. This is some of the team, Carly and Chris, and behind the camera, Shanzi, who looked after me throughout my treatments. So it was just 8 treatments. I used to go twice each week, each time, to get my bloods done, see the clinic, get my neck checked for any other lumps, and then I'd have the treatment. Basically, the treatment involved taking a whole load of painkillers and having an injection, and then just sitting on the ward for the rest of the day to see how I was doing. I got so good at this, I actually wrote all my Christmas cards that year, sat on the ward. But it was great. You feel a bit like you've got flu, but compared to what I'd had before, this was amazing. In January, I had apheresis, which is where they filter out bits from your blood to go back to the labs. And then I got follow-up checks for the following 3 months. And this really helped me as well. It started to give me confidence back in my body. I can't speak highly enough about the team there. They were great. Oh, and that's part of my fundraising thing. So where am I now? I'm doing what I like to call freestyling. I managed to retire early, and that's my lovely husband, who was my carer throughout. And we're trying to get in as many adventures and travel as we can. One of the reasons I asked, am I going to die, was because of that young man there. That's my son, Callum. And I was just so privileged to be able to attend his wedding to the lovely man on. All of these things have been great for me. I've made friends in Poole, so I'm no longer isolated. I'm a serial volunteer. I enjoy my life. And I'm really grateful to the NHS, first of all, because I have this life. But also grateful to have been able to take part in this trial and contribute in my own little way, one of those little boxes. I'm really proud to have done it, and I really hope that in future it's going to stop other people from having to go through this, and also save the NHS all that cost, all that time that I had to go through. So that's it. That's from me.
Thank you very much, Ali, and to all our speakers, and particularly Ali. The others are paid to be here in one way or the other, but you're here as a genuine volunteer. Well, indirectly, We're going to bring some chairs up, so hopefully our speakers and I can sit in front. We have about 20 minutes, and I'm going to try and alternate questions from online and from within the room. And I'm going to sit on the left, so just simply because that's what I was told to do. And is this on? This is on, great. So I'm going to take a couple of questions from online first, and I also notice you have the facility to upvote questions, which you hopefully can all see. So I'll take these two together from Anonymous. How long does it typically take for a cancer vaccine to go from research to patient use? And then what types of cancers currently have vaccines available or in development? And I'm guessing probably the two of you, but quick answers, please, so we can get lots of questions.
It can be
quick
long.
answers, Gareth.
Right. I think it's about 10 years, from discovery in the laboratory, because you have to go through those phases of trial. You've got to work out the dose, then you've got to do another study to work out whether it's safe and if it's having an effect on the cancer, and then we do those big trials where we compare it to the normal and get sufficient evidence for NICE to approve, because we also have to work out the cost, because they need to know the cost, whether they can do it in NHS. So I'd say about 10 years in that sort of region.
And vaccines in development? Cancers?
Lots. So COVID basically gave us a new technology which is proven, and I'm sure everyone is aware how quickly that went through the regulatory hurdles. So it gave us a huge amount of information and confidence that we could design those things and then put them into people. It was a fantastic test bed, not a great thing at the time, but it was a fantastic test bed for the technology to show that we could deliver those things, and actually we could then understand fighting off a viral infection. We could use those types of technology for then doing it against cancer because of the reasons I mentioned, immunosuppression, immune evasion, all of those things are an extra hurdle that we need to build in. So all the design now is about trying to understand what the tumour is doing and what we need to do extra than what we did with a COVID-type vaccine. But yeah, there are lots of strategies for doing that.
Great. Looking for questions in the room? Judith.
Thank you very much. It was really, really interesting. Two questions. Are you using AI to sort of speed some of this up, and where does it most play its part? And how does the cost compare? I was just picking up on your last point about between the sort of the standard treatment and the sort of vaccine. I suppose you haven't necessarily got far enough to sort of make that kind of comparison, and each case is individual, but in general terms.
AI is increasingly used and it's very useful, although us professors have to do the on-off button, so it can't control us. Because what we do is when we collect samples, we get millions of data items. And, a statistician like me can pick up patterns in the data if there's benefit or things, but you can only deal with a couple of hundred. But AI will deal with hundreds of thousands, millions of pieces of data and start telling you about patterns. So it's very, very important to use. In terms of cost, we know that NICE will generally approve a treatment at something called a cost per quality adjusted life year, which means that what they work out is what is the cost for an additional good quality year of life for an individual for the cost of treatment. And NICE will use about 20,000 to 30,000 as the cut-off. And what they'll say is if it's cheaper than £30,000 per quality adjusted life year, we'll approve it, and you can get on the NHS. But if it's more expensive than that, they often say we can't afford to do it. But that's why it's important when we collect the samples, we send them to our colleagues like Mark, because some patients may benefit more because their genetic profile to a drug. So if we start selecting patients out, you get much more greater benefits, which we saw with HER2 and Herceptin in breast cancer patients. So we're still investigating whether, why are those patients benefiting a lot more to those cancer vaccines and why are those not benefiting so much in the laboratories, so that we can identify them at the beginning and then increase our cost per benefit so it becomes standard of care.
Yeah, just to add to that, one of the things that's really important in cost is actually the duration of response. So if you can cure someone, i.e. they go out 10, 15 years, then obviously that's saving them coming back for further treatment. So one of the really attractive things about immunotherapy is it has this concept, and Esme talked about it a little bit, of inducing memory, which means you've got immune cells patrolling that are directed against that cancer. So they can stop it coming back, which is something that chemotherapy doesn't have. It just kills what's there and then it's out of the system, whereas having memory is the thing that's really exciting, and vaccination is one of the things that we do all the time for all the viruses and bacteria you get immunized against, are inducing memory, so the next time you get it, you get a quicker, better response.
I'm going to jump a couple of questions, be inclusive for Esme and prepare her for her Viva coming up. And these are undoubtedly from aficionados, Esme. So, first of all, from Mina Parsley, is STING agonist function dependent on P53 gene? What happens if there is a P53 mutation?
So, P53 mutations are in the tumour cells, and that's one of the mutations that I believe contributes to the growth of tumour cells and how they become cancer cells. But STING, because it's in our immune cells, it's sort of can act independently of what the tumour is doing, really. So potentially STING agonists are applicable to quite a broad variety of different cancers, sort of regardless of what mutations they have.
Great. I've got another one for you in due course. But Ali, and this is from Professor Rosie Fluid. How is it possible, or maybe it should be, what was your route for a cancer sufferer to become a volunteer?
I had a long career in marketing, which is basically persuading other people to buy stuff that you sell. And my husband had a different career in social housing, and it was just time, I knew, for me to give back. So it's not just what I've done here. I'm a restorative justice practitioner. I do all sorts of things because it was time for me to give back to society.
Wonderful. Thank you very much. Veronica. Sorry, it's not just people. You can put your hand up if I don't know your name. I'll still.
Thank you. Thank you all for the excellent talks. Are there any of the big pharmaceutical companies involved in all of this, and if so, which ones?
There are a number of companies that worked in COVID to develop RNA vaccine type studies, and we do know, whatever we think about COVID vaccines now, they did save lives, and they use that technology, microRNA vaccines, to develop a lot of these new cancer vaccines. And we have to work with them. They've got the money and the resources to develop a product. But when we run it within the University of Southampton, we're academics. So we will always report the data, whether it works or whether it doesn't work. But we work with the likes of biotech, Moderna, and there's other companies as well. So we spend a lot of time interacting with them to conduct these studies. And that platform that I described, the Cancer Vaccine Launchpad, we've said to the whole world, if you are a company and you're investigating cancer vaccines, come and talk to UK PLC, because we can go out to the patient population, and any patient that wants to come into a study, we can direct them into your study to get those state-of-the-art cancer vaccines.
Another question in the room? Yes, sir, in blue up there.
Earlier on, there was a graph, the STING, the amount of immunisation, I suppose you call it, against how effective it was. And you said if it was, you put more in, it was less effective. But you didn't show what happened if there was less put in.
Esme.
Yeah, so less would typically also lead to less of a response. So we sort of see like a bell-shaped response, and it's quite difficult in patients to kind of identify that little spot in the middle where you're going to get a good response.
So a couple online, one from Patrick Waters. Are the vaccines targeting precancerous cells or actual cancer cells?
They can do both. If you know that, for instance, if there was a patient that had a particular familial hereditary mutation, you could target that, knowing it was there, and that's before someone had actually generated cancerous cells. So you could definitely do that. But generally, like with the cancer vaccine launchpad, what you're looking at is the mutations that are actually in the tumour. But the complexity of cancer is such that not all cancer cells are the same and identical. They're genetically quite diverse. It basically becomes a game of Darwinian evolution, of selection across the immune system, attacking all of those tumor cells, and there being a population that maybe evade the immune system. And that's where the vaccination comes in. It's actually knowing which mutations might be most helpful for generating an immune response against. But that could be, indeed, precancerous, postcancerous, in the adjuvant setting, in the neoadjuvant setting.
There was a question, a gentleman at the back row. Could you use the microphone, just so that people online can hear? The 500 people online, I'm told.
Thank you. I just wanted to ask where we stand on the treatment for bladder cancer. I was nearly going on a trial with that, but for various reasons that hasn't come available. But I just wondered if this is following the same logic, or if each cancer is different and you need a different strategy.
Each cancer is different, and even in bladder cancer, you'll get different types of cancer cells that are causing the bladder cancer. There's very clear evidence that immunotherapy works in bladder cancer now, so we know that, and we're drilling down to work out which patients are benefiting. And they're definitely developing new cancer vaccines to target bladder cancer. I think what happened is a lot of the big pharmaceutical companies and biotech have identified things like colorectal, because as you saw, I mentioned there's 1.2 million people with colorectal cancer. So companies often start in the big cancers first, because they have an economical model. And then what us scientists do is we approach them and say, can we use your cancer vaccine and experiment and do trials in other cancers that you're not targeting. But there's certainly, you'll see cancer vaccines already to start to be used in bladder cancer.
And there's a question online from Andrew Simon about pancreatic cancers.
Pancreatic cancer as well. Yeah, all the cancers are being investigated currently.
Okay. Thank you. A question online from Jay Buckingham. How could cancer vaccines complement existing immunotherapies, e.g. ABS, potential for combination therapies?
I like that question, thank you. ABS is
Planted
antibodies. Yeah, not planted, no relatives, no changed names as far as I'm aware. That's exactly where we would imagine that we're going to go. So the vaccination to a mutation that's in a tumour is the bit that gives you the specificity. Antibodies can do the kind of thing that STING agonists can do, that Esme was talking about, which is provide that adjuvant effect. It stimulates the immune system to get bigger responses. And the challenge for us is to work out which antibodies to which receptors on which immune cells are the best ways of going ahead. And as Gareth just mentioned, that might be different for different types of cancer. Bladder cancer immune suppression is probably different than pancreatic, different than breast, etc. So that's the challenge for us, to understand how to use these different combinations in the most effective way.
Great. Yes, please. And just if you just wait a second for the microphone to reach you.
Okay. With all this vaccine, are you able to, or potentially cure brain cancer, or will the vaccines not get into the brain?
That's a really good question. So you do get some immune cells into the brain. The question is whether that's always very helpful. I don't know if you're aware of a different type of therapeutic immunotherapy called CAR T cells. So there what we do is we take one of the immune cells in your body, called a T cell, which is, as we mentioned, can go off and then kill cancer cells. You give it almost like an antibody in its surface, so it will go and find the right type of cancer. And in blood cancer, we show that those things can work very well. But occasionally what happens is that they'll get into the brain and cause some toxicity. So we have to be very careful in the brain. It's immune privilege. You don't normally get most immune cells in there. But that doesn't mean that that isn't a strategy that you could use. We just have to be very careful when we're developing those kind of things. Immunotherapy in general, you can use immunotherapy antibodies to boost responses that will clear brain cancers in preclinical models. People haven't yet demonstrated that in the clinic. Yep.
Gentleman on the middle row over there, please.
And can I just ask about prostate cancer, since that's one of the other major cancers, what progress is happening there?
I think, again, in immunotherapy, a lot of progress in immunotherapy. They've actually recently had practice change in studies with immunotherapy, and that will, done by others, that will change practice now going forward. The beauty of that cancer vaccine launchpad is we started with colorectal, and we sent a message out to the world, we are looking for other studies. Until they sign the contract, I'm not allowed to say, which disease, which types of cancer, which companies, but they're actively adding on all cancers to these cancer vaccine launchpad investigations. But it takes time, because if you get a company that's developing their vaccines, what they ultimately have to do is they have to get licensing with the FDA, Food and Drug Administration in America, the MHRA within the UK. And they have to do studies where they get clear evidence, done in a very regulated way, a clear costing. So a lot of these companies generally go, well, I go for licensing in colorectal, I'll go in breast, because the market is big. And as soon as it gets licensing in those big cancer areas, you start expanding it out, the license, to cover other areas. Or researchers like us go to the company and said, well, all we want is your cancer vaccine. Don't give us any money. We'll get that from Cancer Research UK or the Medical Research Council. So you'll find this sort of cascade of all these new cancers investigating the vaccines. So it's inevitable that they're going to investigate in all those cancers.
And the lady just along.
Thank you. Is the hope or expectation that immunology will in time replace the need for chemotherapy and radiotherapy, or is it envisaged that it will have to be used alongside?
It will replace everything. No, sorry.
When I was young and confident.
In all seriousness, if you look at the kind of history of any kind of scientific discipline, you go through these things where we didn't understand something, someone tried something, it worked, we didn't really understand why, and we thought we understood why, and then we're going to go through these cycles. But I think when you look at the way that the sophisticated technology we have to understand what's going on, that gives us the potential to understand in every individual how we might treat that patient. So I think using immunology, it makes perfect sense that you should at some point be able to do that. When that will be, how long that will take, is something that we can't say. And the whole kind of discussion around AI, I think is sometimes a little bit oversold in terms of what it can do now, but in terms of what it could do, in terms of, like Gareth said, looking at large patterns, large data, the more data we collect, the more data rich it is, allows you to understand why, those 10 patients responded and those 12 didn't. That kind of level of data, that then allows us to then think, okay, how do we circumvent that problem? We design new therapeutics, what type of therapeutics they might be, antibodies, cells, vaccination, all of those things in combination. But I think you get to a point where you can understand why that tumor has escaped the immune system. You can circumvent it with new therapies that can reinstall that. So conceptually, I think that's a reasonable expectation. But like I say, how long that will take us to get to is what we can't say.
We're right at the end of time, so I'm just going to ask a couple of questions from the online group, and then we'll conclude. And I'll lump these together. So first of all, from Joe Alcock, I'm a medical student. How can healthcare professionals facilitate research such as these into practice? And from Domenico Radici, for Esme, I suspect, why would you want a higher dose of STING agonist if the lower dose achieves a higher rate of T-cell activation? Medical student involvement and...
Yeah, so that is a good question. It's kind of an obvious question, I think. One of the challenges is identifying what dose is going to lead to what response. And so one of the kind of challenges of all immunotherapy really is that you start to get adverse responses with sort of repeated doses or higher doses. So I sort of didn't have time to go into that much, but what I'm also hoping is that those combination treatments can reduce the adverse effects of higher dose agonists. So potentially we're sort of able to increase the window of dose which can be given and still have benefit in patients. So rather than kind of trying to hit this really small window that might be quite difficult to induce an immune response, we're kind of able to induce better efficacy in more patients with that option.
And healthcare professional involvement?
So in terms of clinical trials that will change practice, the most important group are the patients, because if they don't come into the studies, we can't get the evidence. The second, in my view, most important are the doctors, the NHS researchers in the hospitals talking to the patients and recruiting them into studies. Each hospital in the trial has to have a PI, the main person responsible. But if you're a young and up-and-coming clinician that wants to get into research, you can register as an associate principal investigator, which means you work with the researchers in that hospital to help identify patients to come into studies. And I would encourage, those medical professions, don't stop doing research until we've cured everything in the world, and you need to have that, psyche of always doing research, and there'll always be opportunities in your hospital to get involved, interacting with the patients, because you're the one seeing them and recruiting them into studies. So that's what I'll say for the younger generation of medics.
Great, thank you. My screen went blank ominously, which tells me we've reached the end of our time. Hopefully our online audience is still with us. I'd like us all to just thank our speakers, particularly for their talks, but then how much they engaged in the question and answer session we've just had. So please.